Hello students, in this session of chapter number 4, that is molecular basis of inheritance, we are going to learn the topic that is semi-conservative method of DNA replication. In 11th standard, you have already studied the detailed structure of DNA. So DNA during the process of cell division produces exact copies of itself or it duplicates. This process is called as DNA replication. So we are going to learn the process of DNA replication in stepwise manner. DNA replication is the process in which DNA duplicates or DNA produce exact copies of itself during the process of cell division. Now DNA molecule regulates and controls all the activities of the cell. So cell carry different activities like protein synthesis, then transportation of nutrients from one cell to another cell. So all these activities are controlled by DNA. Hence DNA plays important role in all the cellular activities. Now during the process of cell division, DNA duplicates and gets distributed equally into the newly formed daughter cells. Then you know that DNA is the genetic material found in most of the organisms. Genetic material carries genetic information from one generation to another generation. So as a carrier of genetic information, DNA performs two functions. The first one is heterocatalytic function and the second one is autocatalytic function. Now in heterocatalytic functions, DNA directs the synthesis of chemical molecules other than itself. For example, transcription and translation. So these processes like transcription and translation are directed or they are under the control of DNA. Now in transcription, there is the production or synthesis of RNA on one of the DNA strand. Then in translation, translation means it is the expression of DNA, that is in the process of translation along with RNA, DNA produces different kinds of proteins which are essential to the cell and the process is called as translation or protein synthesis. Then in autocatalytic function, DNA directs the synthesis of DNA itself. An example of it is replication of DNA. So the autocatalytic function carried by DNA is the process of DNA replication. Now in the process of DNA replication, DNA produces exact copies of itself or duplicates to produce new DNA molecules. Now the process of replication occurs in different steps. So we are going to learn this process in a stepwise manner. Before studying the process of DNA replication, in short, we are going to revise the Watson and Crick model of DNA. Now, DNA molecule consists of two anti-parallel polynucleotide strands. These two strands cross with each other, hence they are called as anti-parallel strands. Then these two strands are held together with the help of weak hydrogen bonds. Here we can see that these are the two strands and these two strands are held together with the help of weak hydrogen bonds between the nitrogen bases on the opposite strand. Then each strand consists of a polynucleotide chain. Now polynucleotide chain means number of nucleotides. Here we can see a nucleotide. Then here another one. So these nucleotides are linked with each other with the help of phosphodiester bonds. Then each strand shows two ends, that is C5 end and C3 end. C3 end means the strand ends with third carbon atom with free hydroxyl group, and C5 end means the strand ends on the fifth carbon atom with free phosphate group. So hence each strand shows two ends, that is C5 and C3 end. Then the purine and pyrimidine ratio on both the strand is 1:1. Here we can see adenine pairs with thymine with two weak hydrogen bonds, while guanine pairs with cytosine with three weak hydrogen bonds. So the DNA molecule consists of two antiparallel polynucleotide strands. Then in a single nucleotide we can see sugar molecule, phosphate group and one nitrogen base is called as nucleotide. Then the dimensions of DNA molecule. The diameter or the width of DNA molecule is about 2 nanometer or 20 Armstrong unit. Then the distance between two nucleotide is 0.34 nanometer, or in the form of or in the conversion of Armstrong we can call it as 3.4 Armstrong unit. Then the distance occupied by each complete spiral, that is from here to here. So this distance occupied by each complete spiral is 3.4 nanometer, that is 34 Armstrong unit. So this DNA molecule in the process of replication produces exact copies of itself. Hence the process is called as replication of DNA. Now we are going to learn the process of DNA replication. We are going to learn the process of DNA replication in eukaryotes. So in eukaryotic organisms, the replication of DNA occurs only once in the cell cycle in the S phase of interphase. You have studied the details of cell cycle and cell division in 11th standard. So during the S phase of interphase, the replication of DNA occurs, and the replication of DNA occurs through the semi-conservative method or semi-conservative mode of replication. The process involves different steps. The first one is the activation of nucleotides inside the nucleoplasm. Then second one is the point where replication starts. It's called as point of origin. Then the third one is the two strands separate from each other, that is unwinding of DNA molecule. Then the fourth step is formation of Y-shaped structure after the unwinding of DNA strand, and it is called as replicating form. Then on each separated strands, new strands are produced, and on these separated parent DNA strands, new strands are produced. These are leading and lagging strands. Then finally, in the seventh step, there is formation of daughter DNA molecules. That is, from one DNA molecule, two daughter DNA molecules are produced. Now the first step in the process of DNA replication is activation of nucleotides. Four types of nucleotides of DNA, that is deoxyribonucleotide, adenine monophosphate, deoxyribonucleotide, guanine monophosphate, then deoxyribonucleotide, cytosine monophosphate, and deoxyribonucleotide, thymine monophosphate are present in the nucleoplasm of the nucleus. So nucleoplasm is present inside the nucleus. Then these nucleotides are activated with the help of ATP, that is adenosine triphosphate. ATP are rich, or they are energy rich molecules. So with the help of ATP, in presence of enzyme phosphorylase, these nucleotides get activated and the formation of deoxyribonucleotide triphosphates occurs, and these triphosphates produced are DATP, means deoxyribonucleotide adenine triphosphate, then deoxyribonucleotide guanine triphosphate, then deoxyribonucleotide cytosine triphosphate, and deoxyribonucleotide thymine triphosphates are produced. As ATP is used for the activation of nucleotides, the process is called as phosphorylation. That is, phosphate is added, hence it is called as phosphorylated. And these activated nucleotides are used to produce a long chain or a polynucleotide chain of nucleotides, and resulting in the formation of a new strand. Now the second important step in the process of replication is that the process starts at a specific point on a DNA molecule. It is called as point of origin. So the process of replication start at a specific point on a DNA molecule called as point of origin, or it is also called as O point, and the process ends at a point, it is called as T point or termination point. Now here we see a DNA molecule, and in the next diagram we see that these two strands get separated from each other by cutting the weak hydrogen bonds between the two strands. So the unit of DNA in which replication occurs is called as a replicon, and this point where the process starts and the two strands start to separate from each other is called as origin point, or it is also called as starting point or point of initiation, or in short we can call it as O point. At this O point, the enzyme endonuclease cuts one of the DNA strand temporarily by breaking the phosphodiester bonds. Then in the last diagram we see that the two strands of DNA molecule start to separate from each other, and simultaneously two new strands, that is one on each strand, is produced or synthesized. So these details we are going to see in our next point in the process of DNA replication. Now the third step in the process of DNA replication is unwinding of DNA molecule. We have seen that the process of replication starts at a specific point. It is called as point O, then point of initiation, or it is also called as starting point. So at this point O, the enzyme called as DNA helicase breaks the hydrogen bonds between the two strands near point O. Here in this figure or diagram you can see that the two DNA strands have separated from each other by breaking weak hydrogen bonds between them. Then these two strands separate and unwind. They separate from each other and form a Y-shaped structure. Here we can see that this is a Y-shaped structure, and this Y-shaped structure is called as a replicating fork. Then each separated strand, that is this one and this one, act as a template or the site on which new DNA strand is produced. Hence it is called as template strand. And during the process of replication, these two DNA strands get separated from each other, but sometimes they may again rejoin with each other. So to prevent these strands from joining, single-stranded binding proteins, or in short they are called as SSBP, are present in the nucleoplasm. So these single-stranded or single-strand binding proteins prevent these two strands from joining to each other. So the two strands get separated in this step, that is unwinding of DNA molecule with the help of enzyme DNA helicase at the point of origin. Now the fourth step in the process of DNA replication is formation of a replicating fork. We have seen in the previous step that the unwinding of two strands occur, and due to this unwinding and separation of the two strands, a Y-shaped structure is formed, and it is called as a replicating fork. Here we can see in this diagram that the two strands have separated from each other, and due to the separation of these two strands, a Y-shaped structure is formed, and this Y-shaped structure is called as a replicating fork. And due to the unwinding of these two strands, a strain occurs on both the strands due to the separation, and this strain is relieved by an enzyme called as super helix relaxing enzyme, so that the strands do not break or do not cut. So the strain is relieved by super helix relaxing enzyme, and on these two separated strands later on new strands are produced. Here we can see that a new strand is being produced and another one is being produced here. So a replicating fork is formed due to the unwinding of DNA strands of the parent DNA. Now, after the formation of replicating fork, production or synthesis of new strands occur. So each separated strand, after unwinding, acts as a template for the synthesis of new complementary strand. Here we can see that these two strands of DNA molecule get separated at point of origin, and each separated strand act as a template for producing new complementary strand. Then RNA primer molecule get attached to C3 end of the template strand and attracts complementary nucleotides from the nucleoplasm. Now here we see that at this template there is C3 end. Then on opposite template C3 end is here. So at this end the RNA, that is C3 end of the template strand, the RNA primer molecule gets attached. Then here at the C3 end of this template, RNA primer gets attached. And this RNA primer brings complementary nucleotides, means the nucleotides which are present in the nucleoplasm. It brings these complementary nucleotides from the nucleoplasm to the site of the replication. Then these nucleotides bind to the complementary nucleotide on template strand, that is these template strand. So the new nucleotides, these are the new nucleotides, bind to the complementary nucleotide present on the template strand. And these nucleotides get bind with the help of weak hydrogen bonds. Now adenine pairs or bonds with thymine with two weak hydrogen bonds, while cytosine pairs while guanine pairs with cytosine with three weak hydrogen bonds. So these complementary nucleotide get attached to the nucleotides on the template strand by weak hydrogen bonds. Here they have not shown the actual, that is three weak hydrogen bonds are not shown here in this diagram, but guanine pairs with cytosine with three weak hydrogen bonds. Then these newly bound nucleotides to the leading template, and these nucleotides also bind to each other with the help of phosphodiester bonds. So these nucleotides, here we can see this nucleotide, then second, then third. So these nucleotides bind with each other with the help of a bond called as phosphodiester bond and produced a long chain of nucleotides called as polynucleotide strand. So the synthesis of these new complementary strands is catalyzed by an enzyme called as DNA polymerase. So DNA polymerase catalyzes the synthesis of these new strand on the old or template strand. So always complementary strand is formed in C5, here we can see that C5 to C3 direction. Then on opposite side we can see that the complementary strand is formed from C5 to C3 direction. So in this way, after the unwinding of DNA molecule, two new strands are produced on the old strands, or they are called as template strands. During the synthesis of new strands, the two strands produced are called as leading and lagging strand. The template strand with C3 end is called as leading template, and the template strand with free C5 end is called as lagging template. Now here we can see that the template strand with C3 end, it is called as leading template, and the template strand with the free C5 end is called as lagging template. Then the replication starts at the C3 end of the template strand and proceed towards the C5 end. Here we can see that the replication starts at the C3 end and it proceeds towards the C5 end. Here also we can see the replication starts at the C3 end and proceed towards the C5 end. One new synthesized strand develops continuously. Here we can see this strand develops continuously towards the replicating fork. Replicating fork is here. So this strand which develops towards the replicating fork and it is continuous, it's called as leading strand. Then another strand develops away from the replicating fork. Here is the replicating fork and another strand we see. It develops away from the replicating fork and it is discontinuous, or it shows presence of DNA fragments. Such strand which develops away from the replicating fork is called as lagging strand. So during the formation or during the process of DNA replication, leading and lagging strands are produced. DNA synthesis on the lagging strand forms, or it is produced in the form of small fragments called as Okazaki fragments. Now, as the scientist called Okazaki first discovered these DNA fragments, these are also called as Okazaki fragments. So here in this diagram we can see that one is leading template and another one is lagging template. On leading template, the strand is produced from C5 to C3 direction towards the replicating fork is continuous. Hence it is called as leading strand. And the another one which is produced or which develops away from the replicating fork from the direction that is C5 to C3 is present in the form of small fragments of DNA, or it is discontinuous. As it is discontinuous, it is called as lagging strand, and the fragments are called as Okazaki fragments. So these Okazaki fragments are joined with the help of an enzyme called as DNA ligase. Then the RNA primers which are attached on the template strand or parental strand at the C3 end. Here the RNA primer was attached. Then at the C3 end of this leading template, RNA primer was attached. So these RNA primers are removed with the help of enzyme called as DNA polymerase. And finally, an enzyme called as DNA gyrase or DNA topoisomerase forms double helix to form daughter DNA molecules. So double helical daughter DNA molecules. In all, two daughter DNA molecules are produced from the parental DNA molecule. The last step in the process of DNA replication is formation of daughter DNA molecules. So at the end of the process of replication, two daughter DNA molecules are formed. So here we can see that two new daughter DNA molecules are being produced. Now in each DNA molecule we can see that one strand is old. Here the old strand is shown in blue color and another one which is new, it's shown in red color. Here also in the another DNA molecule or daughter DNA molecule we see new strand and one is old strand. So in each DNA molecule one strand is old, or it is also called as parental strand, and another strand is totally new, or it is newly synthesized. Thus 50% or the half of the DNA is old, while half of the DNA is new in newly produced two daughter DNA molecule. So the method of DNA replication is called as semi-conservative method of replication. It means that one strand, that is old strand, is conserved, and on this old strand a new strand is produced, and half is old DNA while half is new DNA, and old DNA is partly conserved. The process of DNA replication is called as semi-conservative method of DNA replication. So in this session we have studied in detail the process of DNA replication in eukaryotes. So the remaining topics we will see in our next sessions.